Okay, so now we're going to start talking about how lipids are transported in the blood and what blood lipid levels depend on. So they depend on a number of different factors. One would be age, two, gender, lifestyle, for example, exercise, genetics, and also diet. So of course, what we eat depends on our amount of fat that is going around in our blood. So utilization of fats during exercise, according to Galbo and his colleague, fat combustion, so meaning fat usage, reaches its peak around 60% of our VO2 max. Okay, so when we talk about VO2 max, that's our maximal oxygen consumption in a given period of time. So how much are our muscles and our body uptaking oxygen and utilizing at any given point in time? So when we reach about 60% of that, we are mainly using fats. Okay, so right here, fat combustion, we see, it gives us 4 kcals per minute here. Right here at about, again, these are all lining up about 60% of our VO2 max, right? Fat use also increases as the duration of exercise increases and with training. So we can become better fat burners as we train, and that's what we want to do. We want to spare glycogen. For example, let's use a marathon example. If I want to run, I want to use our most efficient fuel to give me the most ATP to keep exercising, correct? If I use my glycogen stores and glucose stores too quickly, I'm done. I'm pretty much out. And if I don't know how to access my fat stores, that exercise task is pretty much going to stop. So with training, we actually become better fat burners. And what we call is we engage in glycogen sparing techniques. And that's ultimately what occurs with training. All right, so the rate of fatty acid oxidation depends on a number of different things. One, our exercise intensity. So how intense are we exercising? Our training status. Are we trained versus untrained? So like I just said, we're become better fat burners and we know how to access our fat better metabolically with training. Plasma free fatty acid concentrations and endogenous and exogenous carbohydrate availability. So all of that affects our rate of fatty oxidation metabolism. Okay, so continuing to talk about utilization of fats during exercise, fat contribution to total energy expenditure during exercise. This increases to a high point out of about 50 to 65% VO2 max, and then it decreases as the intensity reaches 85%. Okay. As we approach 85% of our max VO2, lipolysis is not adequate to meet the energy demands of the muscle cell. However, researchers have found that the exercising muscle is also an important site for the control of fatty acid oxidation. All right, so let's talk about intense exercise and fats. And the reason we don't really access fat tissue while we're in high intensity exercises are because of these reasons below. During intense exercise, we talked about last week, the flux of glycolysis is enhanced. So we're going through it very rapidly. This tends to lead to an elevation of pyruvate and acetyl-CoA, where depending on what we're using for glucose at the time, we can either make, again, like we said, pyruvate or acetyl-CoA. But elevation in those levels inhibits the action of carnitine. I'm going to let you figure out that last word, but ultimately it's this carnitine enzyme that we use as an important step in lipid metabolism, and that enzyme is deactivated. So our uptake for our fatty acids by the mitochondria will be then reduced. So ultimately, to reiterate, an elevation of pyruvate and acetyl-CoA during higher intensity exercise inhibits the action of this carnitine enzyme, and therefore uptake of our fatty acids by the mitochondria is reduced. So ultimately, the end of the story is we're not going to be using as much fat tissue during intense exercise. Okay, so there are 7 steps to fat mobilization and fat metabolism ultimately. So fat is divided into the following phases. 1, mobilization, circulation, uptake, activation, translocation, ultimately beta oxidation, and then lastly, oxidative metabolism. Number 7 ultimately is those reducing equivalents going over into acetyl-CoA and NADH and all that going over into the Krebs as well as the ETC. Okay? So let's talk about step 1, mobilization. Where does lipolysis occur, right? Remember we talked about last week that glycolysis occurs in the cytosol of the cell, but Krebs is in the mitochondria, as well as the ETC is along the cristae of the mitochondria. Well, lipolysis occurs in the cytosol of the cell as well. The key enzymes in this step 1, in this process, is in mobilization. 2 enzymes are desnutrin and hormone-sensitive lipase. Desnutrin is the first enzyme to work on the triglyceride, the TAG. This forms a 1,2-diacylglycerol and a free fatty acid. So this is the first enzyme that accesses the triglyceride in order to break it down to use for energy. 2 is hormone-sensitive lipase. This removes the second and third fatty acids from the triglyceride, and this is highly activated by glucagon. So lipoprotein lipase, these remove fatty acids from fats that are attached to triglycerides within lipoproteins within the blood. So remember we were saying there's also, we can access our triglycerides from our fat tissue, like adipose tissue and storage sites, but we also have a given amount of fat floating throughout our bloodstream at any given point in time. And they're usually attached to a lipoprotein. So if you see the enzyme lipoprotein lipase, instead of hormone-sensitive lipase activating those, accessing those, an enzyme that's floating in our bloodstream called lipoprotein lipase is the one that removes fatty acids from fats that are attached to triglycerides that are floating around in our blood. This is synthesized within cells and then transported to the surface of the capillary endothelium. Okay, this is activated by insulin. And highest concentration you will find this is in the heart, within adipose tissue, and also slow twitch red fibers. So let's talk about hormone-sensitive lipase. This is that major enzyme that's accessing the second and third fatty acid from the TAG. This is inhibited by insulin. So a high insulin to glucagon ratio will inhibit the activity of hormone-sensitive lipase. So ultimately, this is why plasma free fatty acids are higher in the fasted state when insulin levels are low than in the fed state. A small amount of insulin is needed to suppress lipolysis. Insulin has a very long-lasting effect on hormone-sensitive lipase, right? So ultimately what it does, we kind of briefly talked about this last week, insulin helps to dephosphorylate hormone-sensitive lipase via protein phosphatase. You don't need to ultimately know that last section of it, but ultimately insulin will remove a phosphate and deactivate hormone-sensitive lipase. So now, ultimately, what can't we do? If hormone-sensitive lipase can't work, we can't access our triglyceride stores to break them down for fat. So that's what we started talking about a little bit last week with regards to a high carbohydrate diet or eating a lot of sugar or so forth. That, for the most part, gets broken down into glucose. High amounts of glucose in the bloodstream signals our pancreas to release a large amount of insulin. The more insulin we have in the bloodstream, the more it's going to deactivate hormone-sensitive lipase. So that's why, technically, you're not going to be accessing your fat stores as efficiently as when you have so much insulin in the bloodstream as compared to when you have low levels of insulin in the bloodstream. Okay, here, so we're going to talk about lipoprotein lipase versus hormone protein lipase or hormone-sensitive lipase. So here we go. Hormone-sensitive lipase is where we access our triglycerides, right? So we're accessing our fat tissues from our adipocytes. This is an adipocyte over here. And then over here we have our lipoprotein lipase. This is our bloodstream. And they're usually attached to, like we said, a lipoprotein or triglycerides that are floating within the blood. So lipoprotein lipase will access the triglycerides that are floating in the blood, and then we'll break it down to use for energy. But hormone-sensitive lipase is a lot more active in our adipocytes over here. And this is how we activate lipolysis through our adipocytes, and we will produce a byproduct of glycerol. So we will explain a little bit more about glycerol in the next few slides. Okay, so hormone-sensitive lipase. This is stimulated by catecholamines. What are catecholamines? If anyone doesn't know what they are, those are epinephrine and norepinephrine. Those are released from our adrenal medulla of our adrenal glands. And our adrenal glands sit where? They sit on top of our kidneys. Also, human growth hormone has a slow response to activate human hormone-sensitive lipase, as well as cortisol, and glucagon also has a small effect. Okay, so ultimately this diagram over here is pretty much showing you the different pathways of how epinephrine and so forth binding to certain receptors on our cell activates all these different sort of pathways, and ultimately hormone sensitive lipase is allowed to access our triglyceride, and we break it down into free fatty acids and a glycerol, and that's why we will have an increase in glycerol within our bloodstream. We will see an increase in concentration of glycerol with lipolysis. Okay, so this would be considered a adipocyte here, and it's ultimately showing you all the different pathways of what's happening in order for us to activate hormone-sensitive lipase in us to access the triglyceride, split it into the 3 fatty acids, as well as the glycerol backbone. Okay, hormone-sensitive lipase, again, there's a hierarchy of sensitivity to the action of this with catecholamines by the cells. Ultimately, intra-abdominal is first, then abdominal subcutaneous, and femoral subcutaneous. That ultimately is the hierarchy of sensitivity to this by our catecholamines. So technically, we start using, with those catecholamines released, we will usually use intra-abdominal fat stores first. But this is our hierarchy over here. Right, so the release of fatty acids is a high form of intra-abdominal fat depots, but ultimately these fatty acids then travel to the liver. It's believed that this is related to the adverse effects of abdominal obesity. Most fatty acids that are sent to skeletal muscle come from abdominal subcutaneous adipocytes. So that's important to know. Even though there's a hierarchy, most fatty acids that are sent to skeletal muscle in order for us to use for energy, for muscular contractions, we use mainly our abdominal subcutaneous adipocytes to go to our muscle cells. All right, so let's talk about caffeine for a second, as I sit here with my coffee talking to you all about lipolysis today. Caffeine is thought to facilitate fatty acid mobilization by inhibiting the breakdown of what's called CAMP by an enzyme called phosphodiesterase. You don't ultimately really need to know all of those intricacies right there. But according to Graham and colleagues, this only occurs really at the pharmacological doses and not really the physiological ones. So ultimately, we would have to inject high amounts of caffeine to ultimately have an effect to stimulate hormone-sensitive lipase. So you sitting there with your coffee or your espresso or whatever ultimately isn't going to be enough to stimulate hormone-sensitive lipase. But if you are taking caffeine pills or very, very high amounts of caffeine, this is what has been known to seen in pharmacological studies, that it may increase the activity of hormone-sensitive lipase, which is ultimately accessing our fat stores. So this is where the controversy comes in about those caffeine pills and those things called like hydroxycut and all that. Ultimately, it's just a ton of caffeine to help ultimately increase your heart rate and metabolism, all of this type of stuff in order for you to try to break down fat. But again, there's obviously dangers with that as well, and that's another conversation for another day. Okay, so in our next video, we're going to be talking about the markers for lipolysis. So ultimately, as a result of breaking down all these fatty acids, glycerol levels will rise. So we will talk about that next when you click the next video.